Okay, everybody, this is the fourth in this lecture series for Chapter 3. We've already discussed carbon and how versatile of an atom it is. We've talked about its valence. That was the first lecture for Chapter 3. The second lecture was functional groups. So we covered 7 functional groups. We talked about their properties / characteristics, and we talked about their behavior when they're attached to like a carbon skeleton. The third lecture was all about laying down some vocabulary so that as we talk about each of these macromolecules for the rest of this chapter, we all understand some basic key vocabulary. I will briefly, very briefly, allude to them, but I will not take a lot of time to explain what they are again because we've already done it. So let's get started. So the first macromolecule that we're going to discuss are carbohydrates. So carbs are sugars. They're not exclusively sugars though. There are some carbohydrates that participate in the cell wall of a plant. So that's what gives the plant like that crunch when you eat like a salad or any green vegetable that's coming from the ground. What else do carbs do? Oh, carbs can make or help make up the exoskeleton of certain shellfish and bugs. So when you think about carbs, I don't want you to always associate it with bread or cupcakes or pancakes or whatever. Try to just see them as a molecule that is made out of 3 elements. So carbon, hydrogen, and oxygen. And they can vary in the number of carbons they have. And that would dictate what type of carbohydrate you will get. So as far as making a carbohydrate goes, the atoms are just as I said, CHO, and the monomer for a carbohydrate is called a monosaccharide. So if we do the same type of word breakdown that I do quite often, mono means 1. So 1 right there. And then the back half of this word, so after the prefix mono, you have saccharide. And, excuse me, so we're looking at this back half here with the S included. But that just means sugar, like it's a Latin word. It's not saccharide, of course, but that SACCHAR, that translates to sugar. So when you see the term monosaccharide, think 1 sugar. And I'm going to write the word sugar underneath so that it is forever there. 1 sugar. Monosaccharide. Another way to remember this, if you're not good with the whole Latin or Greek origin thing, because it does take some time to, I guess, like build that vocab up in your mind, or those prefixes and suffixes up in your mind, and root words. But saccharin to me also reminds me of sweet. So I think like 1 sweet thing, and I know I just said don't think of all carbs as sugars, but they are sugars, so maybe it is okay in this circumstance to refer to it as 1 sugar. They call them simple sugars. So if you wanted to make a larger carbohydrate, you would be making something called a polysaccharide. In other words, the polymer of a carbohydrate is called a polysaccharide. So you see that word listed right here. And again, the back half meaning sugar, poly means many, so literally many sugars. Okay, so on this slide, we're just going to focus on monosaccharides. What you should remember, in addition to the things that I just listed in the previous slide, you should remember the ratio of carbon to hydrogen to oxygen for a carbohydrate. So the ratio is a 1 to 2 to 1 ratio. So when I'm saying 1 to 2 to 1, I mean for every one carbon, there's going to be 2 hydrogens and 1 oxygen. That's useful if you maybe are looking at a structure and you counted the number of carbons and you know it is a carbohydrate. You can kind of just fill in the rest how many hydrogens and oxygens should also be present. Let me see. The important sugar that they are referencing here is glucose. So glucose is what we actually use to like harvest immediate energy. And you know that people who are diabetic, they have to check their blood glucose levels. So this is the simplest monosaccharide, and this is the one that we find within ourselves. So the chemical formula for glucose is C6H12O6, 6 carbons, 12 hydrogens, 6 oxygens. And if you do the math really quickly, you'll notice that the glucose follows that 1 to 2 to 1 ratio. So how do we group monosaccharides? We group them by looking at the number of carbons they have in their center. So, and then we also have to remember to look for where the carbonyl groups are. So remember this particular functional group. That oxygen is electronegative, so this is going to help the sugar be water soluble. And yeah, that's it for that. All right, so here's a slide. It has 4 different sugars listed, and I believe they break them up into single ones. So I'm going to go forward. So here's the first one that was listed on that page before, or the slide before. So they have triose, which is a 3-carbon sugar, which you should start to see if you're any good with this whole like word breakdown thing that I am doing, is that the prefixes they describe the atom to a T. So the fact that it's tri means that it's going to have 3 carbons. And the only reason why I'm saying 3 carbons is because we know we are dealing with carbon-based molecules. And then whenever you encounter this ending, OSE, it is a sugar. So I know triose, or triose, however you want to pronounce it, is a 3-carbon sugar. Now, if I take my knowledge about the 1 to 2 to 1 ratio for a monosaccharide, I could very easily go, okay, I know there's 3 carbons, so there has to be double the number of hydrogens, and then the number of carbons should equal the number of oxygens. And that is exactly what we find here in the chemical formula. The structural formula looks like this here. Notice where the carbonyl group is, right there. And that's really it. So the name of this particular molecule, or this particular monosaccharide, is glyceraldehyde. So when we talked about the carbonyl groups, I mentioned ketones and I mentioned aldehydes. Notice that the end of this word is aldehyde. That's because that is what this particular sugar is. So glyceraldehyde is it's an aldehyde, and the functional group belongs to the category of carbonyl molecules. Okay, so they describe what glyceraldehyde is. They say it's an initial breakdown product of glucose in a cell. Remember that glucose is C6H12O6, and so if this molecule is a breakdown of this C6H12O6, then that formula makes perfect sense because it's half of the amount of glucose. All right, pentose. So penta, or pent, like pentagram, pentagon. those things have 5, or they mean 5. So when I talk about a pentagon, I'm talking about a shape with 5 sides. A pentagram would be like a star with 5 points. So the pentose sugar, I know that there's going to be 5 carbons, so pent meaning 5, and there's that ending again that I just told you about, that ose, O-S-E, meaning it's a carbohydrate, it's a sugar. So this particular pentose is a sugar called ribose, and what you need to remember is that ribose combines with another macromolecule that we will talk about later, but the other class of molecules are nucleic acids. So ribose is just literally the sugar that is attached to the nucleic acid for this particular molecule. Okay, so moving right along, now we're looking at hexose, which are 6 carbon sugars. So hexagon, that hexa prefix or hex prefix means 6. And then OSE makes it a carb, a sugar. So there are 2 molecules here, and you'll notice they have the same exact chemical formula. They both have 6 carbons, 12 hydrogens, and 6 oxygens. They both are following that whole rule for monosaccharides where we said that the ratio of carbon to hydrogen and oxygen needed to be 1 to 2 to 1. So what is it that is making these 2 not both be glucose or not both be fructose? And if you're thinking about where the carbonyl group is located, then you are on the right track. So what we see here is that we have an aldehyde on this side. Over here, this is a ketone. Remember the ketone, I gave the analogy of like a lock and a key. So the key has to be able to fit in the middle. Key, ketone, get it? Hopefully, I don't know. So there's one more of these that also has a C6H12O6 formula, and the name of that sugar is called galactose. And we will look at it when we do the milk lab, which is coming up. So why are these different? Again, it's just because of where the functional group is located within the carbon skeleton. Glucose is our quick energy source. Fructose is it comes from fruit, so it's sugar from fruit. And both of them can be used for a source of energy to drive you to keep going. Okay, so we just got finished looking at a couple of different linear monosaccharides. What you need to know is that monosaccharides are not necessarily going to be linear. Most of the time they're going to form a ring structure. It really just depends on the environment that they're in. So they would serve as the solute. In this case, if we said the solvent was water, meaning like the sugar was in an aqueous solution, you would get the ring formation. And it has to deal with the fact that there are hydrogens, oxygens present in solution with this sugar molecule. There are a lot of places where maybe some dissociation may occur, specifically when we're talking about that carbonyl group. All right, so here's the linear ring. This is glucose. How do I know it's glucose? Because there are 6 carbons, there's 12 hydrogens, there are 6 oxygens. I don't need to count them because I know the ratio. This slide is just showing you how you would go from a linear ring into, not a linear ring, how you would go from a linear monosaccharide to a ring formation. So yeah, that's it. This is what it looks like. And they have highlighted for you in 2 different colors where the bonding is actually occurring. So you have something happening at the first carbon with the carbonyl group there, and then you have something occurring at the 5th carbon. There's a hydroxyl group there. Just follow the reaction through, and really that's it for this slide. There's not too much more for you to have to dig into, but they just want to show you how the ring formation is possible. Okay, on this slide, there is a little bit more vocab for you guys, so let's test your word breakdown skills. So the important word that's bolded first is disaccharide. Di means two, and then again that saccharide portion of the word is referring to sugar. So a disaccharide is nothing more than two monosaccharides joined together. How do we join them together? What has to happen is a dehydration synthesis reaction. And I'm saying the words like that because I want you to really think about what each one of those words mean individually. If you can understand each of them individually, you can come up with a working explanation of what dehydration synthesis is. So dehydration, to lose the water, and then synthesize means to like make. So, and then the reaction part means it's a chemical reaction that's going to happen. So you're going to take one glucose and maybe a second glucose, and when you put them together, there's some H2O that will leave the molecule, and then you end up with this bond that has an oxygen right in the center of the two monosaccharides. So the linkage that they form, they call it a glycosidic linkage. So let's look at it now.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugarcane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of two linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
Okay, so the next two slides are nothing more than like still frames of the reactions that we just witnessed in the animation. So I'm not going to spend time explaining this, but feel free to pause it, look at it. You should be able to spot fairly quickly where the oxygens and the hydrogens are coming from to make this dehydration synthesis happen. All right, so here we see the end of that reaction, what we get out of it. So the reactants and the products are on the screen right now, and they're drawn for you very nicely. Everything is labeled. You can see the glycosidic linkage that happened in between the two. So we're looking at this area right here. Notice that the carbons are still numbered. Feel free to go back to the other slide where they show you the numbering of those carbons if you wanted to keep track that way, or just to, you know, familiarize yourself with how the count works. Or you could, I guess, have your book open, your e-textbook open, and be looking at it while we're going through this. Okay, so now we're going to talk about the polysaccharide part of this. The polymer of a carbohydrate molecule is called a polysaccharide. I've already done this with you guys, so you should be good to go. What you need to know is that polysaccharides can work to like store sugars or store like that immediate quick boost of energy that someone may need. In addition to that, they can play a structural role within a cell or you know, within any organism. So the way that we determine what the function of the polysaccharide is, we have to look at the structure of the polysaccharide. So whatever sugars are used and the position where the linkages are happening, that's how we can determine function. So structure and function was something that we talked about in chapter 1. Here it is again. This is an emergent property. Like, all of these things are still very valid. So let's keep going. All right, so let's talk about some of the different storage molecules that are polysaccharides. One of them that you should be very familiar with is starch. So starch is just a polysaccharide that is built up in a plant, and it's made out of strictly like glucose. So every time we get this glucose monomer or monosaccharide and we join it with another monosaccharide of glucose, you end up with a really, really long chain the more and more you add them on, and so therefore you get starch. So you think about, okay, well, where is starch? Potatoes test positive for starches. The test or indicator that you may be familiar with using is called iodine, and in the presence of a starch, iodine will turn black. Normally it has like a kind of like a yellow tint or hue to it. So yeah, that's just testing for starches. Starchy foods tend to be things that are like bread-like. So why does a plant need starch? Well, it needs to have like a source of energy stored for itself. We tend to think of plants only performing photosynthesis, and so like therefore why would they need any storage molecules if they are soaking up the sun and making all of this sugar? And the reason is because plants also can do some cellular respiration. So they can do the same process that we can do. They do it a lot more when they are like a seedling or, just beginning to sprout. There's no light involved in that reaction. That's all darkness because you don't put a seed like on top of the ground. You usually put it in the ground. So with the absence of the sun, you have to figure there's something else that is going on that makes this plant continue to grow. So starch is really, really good strictly for storage within a plant. And then when we eat those plants, we are eating their energy store, their starch. So we have enzymes, which are proteins that speed up chemical reactions. And some certain enzymes that we have, they can hydrolyze plant starch. This should say starch. So I found an error in one of the slides given to me by the textbook. Hold on. Ooh, that's not a good color. ST. All right, so enzymes that can hydrolyze plant starch make glucose available as a nutrient. So again, hydrolysis is just the splitting of a polymer. So if the plant does photosynthesis, it makes very, very long chains of these monosaccharides of glucose, forming starch. It's kind of like storing it, waiting for, no pun intended, like the rainy day to come where the sun maybe is not shining, or for some reason there's some cellular respiration versus photosynthesis that needs to happen. You have to be able to break the starch apart, because remember, the energy is in the bonds, and in order to get the energy, we have to break the bond. So hydrolysis is necessary. All right, now let's talk about us. We do not store our excess carbohydrate polysaccharides as starch. We store it as something called glycogen. So glycogen is a storage polysaccharide in animals, so that fits our particular kingdom that we're found in. And then we usually store ours in our liver and our muscle cells. And I think that's appropriate, because if you think about what the job of a muscle cell is, it involves essentially doing some type of work. And you need energy in order to carry out work. So you need to have some type of fuel reserve that you can tap into if you don't have carbs readily available to just like eat and break down right away. So there is the explanation for that.
Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
So hands down, that was the shortest animation ever. I don't know why I was expecting it to be a little bit longer. But they just did the reaction very quickly. They showed you dehydration synthesis, taking all of those different monosaccharides and then putting them together. They form these glycosidic linkages, and then here we are at the next slide. So in this one, we're looking at a couple of different things. First, we're looking at a human who is taking potatoes out of the ground. And so why do they use this picture? Well, because the starch, which is the polysaccharide storage molecule for plants, is found in potatoes, like I said. And then by showing us, there's muscle tissue that's involved in virtually every part of your body. And so with us, we do a glycogen store. So this is just kind of comparing what an animal's polysaccharide storage molecule would look like. So up here, and then for us, here's ours down here. And they don't seem to be that much different. They're still made out of the same exact elements, carbon, hydrogen, and oxygen. It's just the way that the branching happens is different in starch from glycogen. And then in this very last one, we're looking at a plant cell wall. And I think I did already say this, but that's what gives like vegetables their crunch, and also fruits their crunch. And so the plant cell wall is actually surrounded by something called cellulose, which OSE is a sugar, right? So cellulose is it's a carbohydrate, and it's made for like the structure of a plant to make it stand upright, so it's not flimsy and falling over. So here is the potato starch up close. Take a second and look at it, pause it, come back to it. I'm going to keep going. So here we go again with the potatoes. This time we're looking at something called starch granules in a potato tuber. So what you may notice is that if you leave a potato out for long enough, it'll start to kind of bud. And essentially what it's trying to do is like replicate itself. And in order to do that, it has to have some type of energy storage. So the starch is what is going to fuel that process to be able to happen. All right, there's the glycogen in humans. Notice that ours has some extensive branching going on, but it's the same exact type of macromolecule that is making this structure. And then this would be the last one here with the cellulose, making the cell walls, giving plants and fruit that crunch when you eat it. Notice that cellulose is actually, it's unbranched. So there's a difference in the starch and the cellulose, even though both of them belong to plants. So here's the plant cell wall up close. We will be doing some microscopy work. That just means like microscope work very soon. So you'll get to see some of this up close. Same thing with the cellulose, but I don't believe our microscopes at Blair are that great. So this is actually a really nice photo. All right, so now let's just talk about the structural polysaccharides, so the ones that help to form the plant. So cellulose, remember I said OSE, sugar, carbohydrates. So cellulose is a major component of the tough wall, so the cell wall of plant cells. And they compare it to starch and glycogen, but the difference is there's no branching, and it's also the difference is attributable to some of the ring formation that happens when you compare these three molecules. So let me see if they give us a picture. Okay, they do give us a picture. So here's the difference that they're speaking of. Notice that on the left, there's an alpha sign in front of glucose. So that's alpha glucose. And they're showing you how it goes from alpha glucose in a ring structure to a linear structure. And then there's some reconnection that happens that makes it be beta glucose. And this is sort of like a cis and trans situation. They could also be like enantiomers of each other. Okay, so when we're looking at it up close on this slide, they're just breaking down the very top portion of the diagram or chemical reaction layout that we just looked at. And they definitely are enantiomers. If you were to lay these two on top of each other, it's kind of like the situation I was explaining with, your hands not being identical, they're mirror images. Also, notice that there is a hydroxyl group here. So that's like, if you could imagine this being in 3D space, that OH is pointed down and the H is pointed up. Over here with the beta glucose, you see the opposite happening. And remember, the cis-trans situation was nothing more than like, it was a carbon, you had some bonding that was happening, and then it was like a hydrogen here and a hydrogen here would make it cis, and some other designation at the bottom. But if you were to have it like this, where you have one hydrogen up high and one down low, that's the transformation. So that was really, really like sloppy, but I think you get the picture. I'm sorry. All right, here it is once it's all linked together. Look at the position of all of the hydroxyl groups. Notice the glycosidic linkage is between the first and the fourth carbon. All right, and this is in cellulose. So look at this one. This one is a little different because you're linking beta glucose monomers together. You get a low hydroxyl and a high hydroxyl, a low one and a high one. And it repeats over and over again. So this very, very small distinction between whether or not you're bonding all alpha glucoses or if you're bonding an alpha and a beta or it's just straight up beta glucose can give you a very, very different molecule. And the taste of it would be different. The texture would be different. Just like all these little intricacies would be different simply because of the way that atoms are placed when they join each other. Okay, so it's saying that this is like a summary of what we just talked about. So in starch, glucose monomers are arranged in the alpha conformation. So that's starch. And you should also notice this. They talk about how starch and glycogen, they are both helical. So them being a helix just simply means they have like a structure that kind of like goes like that. And along those lines would be all of the monosaccharides that have been joined together with their glycosidic linkage. So cellulose, on the other hand, though, is dealing primarily with the beta glucose conformation. Cellulose molecules are straight because of this. When we did the card sorting activity, there was, I remember like a couple of groups, we did some discussion about the placement of certain atoms and how that would effectively like change the properties of that molecule. This is what we're seeing here, and it's applicable because these are things that you eat, these are things that you're even made of, or things that your body makes as a result of what you eat. Okay, so they reference here the hydroxyl groups on the glucose monomers. They talk about hydrogen bonding that happens between the hydroxyl groups. And then the second bullet point is just saying that when cellulose is made, so if the plant is cranking out like cellulose, it kind of will do this thing like this, right? And they run, so each of these polysaccharides, they're going to run parallel to each other. So parallel cellulose molecules, they're held together, and they're grouped into something called They're grouped into microfibrils. I don't know why I got tongue-tied on that one. So what does that do? Again, it helps the plant with this structure. It's going to make sure that the plant is standing nice and tall when it has enough water. If the turgor pressure is correct, then the cellulose is what is going to help give it that straight up and down kind of a look instead of being wilted. So that's interesting. Okay, so what's interesting about carbohydrates is that our bodies kind of know that they have to be sometimes broken down, sometimes stored. And so we have these enzymes that exist within us that help to make this process of like combining a bunch of monomers together or monosaccharides together to make a polysaccharide and vice versa. But certain species can only digest certain types of carbohydrates. So I'll give you an example. You guys have all heard of fiber, and you know that there's soluble fiber and there's insoluble fiber. So if it's soluble, that means that it can be broken down in an aqueous solution, no problem, harvesting carbon, hydrogen, and oxygen from that. But insoluble fiber is fiber that cannot be broken down in water. And the reason why it cannot be broken down is because you don't have the proper enzymes to break it down. So this is not to say that all species have not all species can break down the same carbohydrate. So they give an example here at the bottom. They talk about cows, and then they are also referencing termites. So they have microbes in their gut that help them to digest the undigestible. So that cellulose that is in a plant cannot naturally be broken down by a cow or a termite. However, the microbes in their gut can break it down. Same thing goes for us. Cellulose in human food, it says passes through the digestive tract as insoluble fiber. So that's where your fiber is from. Your fiber is from the carbohydrates, reinforcing the fact that they're not all sugars and reinforcing the fact that they're not always sweet. All right, so the way that I was taught to pronounce this was chitin. I've heard people call it chitin, but like that just sounds weird. So chitin. So this is a polysaccharide that makes like the exoskeleton of a bug. It also is responsible for shell formation in like crabs. But basically all it is structural support for the cell wall. So they reference here that it can be an exoskeleton in arthropods, and it also can be in the cell wall of like fungus. So that's it for carbohydrates. On the next one, I'm going to talk about lipids.